So today we're here to talk about the effects of bovine respiratory disease on all of our cattle. I've gathered all of us here today because we are the best in the business and have a voice to make the change. This affects all of us in this industry, so does anyone have any thoughts on how to solve this problem?
Yeah, Willem. BRD costs our industry on average $800 to $900 million in economic loss annually. 2.5% of all cattle in the United States are affected by BRD in some way. Morbidity can reach almost 50% in some extreme cases.
There are many different types of treatment, such as non-steroidal anti-inflammatory drugs, and there's also preventative vaccines, but I always see varying responses across my herd.
I agree. My cows are more stressed than ever. They cannot keep up with the necessary daily energy needed to keep them in my herd. They show signs of fever, difficulty breathing, and drooping of the ears, showing signs of sickness. At this rate, I will not be able to afford to make it, and I'll have to cull my whole herd.
There has to be a better way to solve this problem.
What makes the cattle different that have a better response to these vaccines? I wonder if there's a gene that contributes to this. Maybe we can find out how this gene is produced through recombination and use sequencing to determine which cows carry this gene to select to breed.
Now that's an idea that'll work.
In the case of bovine respiratory disease, we have bred out most of our cattle's genetic ability to fight off the virus over the last 100 or so years. However, through recombination, a few cattle have developed a way to genetically respond to the virus better than others. Here's how it works. Recombination involves the alignment of two homologous DNA strands and occurs through complementary base pair pairing. In prophase of meiosis I, homologous chromosomes are aligned on the equator of the cell. While they are lined up, a break occurs at each DNA strand, leaving two free ends. After the break is made, each end infiltrates into the other chromosome, forming a structure called a holiday junction. Finally, after the holiday junction is formed, branch migration occurs. In this step, the holiday junction travels down the chromosome vertically, which results in an exchange of DNA. Through the recombination process, we have created a gene that can more effectively fight against bovine respiratory disease from basically nothing. It is also worth mentioning that animals with this gene are able to easily pass it on to their offspring in a selective breeding program.
Through recombination, we have found genes that produce antibodies that have a unique stalk and knob structure that is able to better deactivate antigens that are a part of the BRD complex.
We can use genome-wide association studies to identify which genes are associated with a particular trait and find genetic markers. Once we have identified the gene, we can use sequencing to determine the specific sequence of nucleotides that make up this gene. One way to do that is through modern Sanger sequencing. First, we have to amplify the DNA segment or gene that we have chosen. Then we will use heat to denature the DNA, which provides two single strands of DNA. We will then attach a primer to the end of a single strand and put that in a test tube. Keep in mind that there will be many strands added into this test tube. We will also add a DNA polymerase and dNTPs that will serve as our genetic material for DNA synthesis. In addition, we add dideoxynucleotides or ddNTPs. These are artificial bases that will stop replication as they are missing a hydroxyl group at the 3' position. This means that no other nucleotide base can attach to them, therefore halting replication. Each nucleotide base will have an equivalent artificial base that has been marked with a specific fluorescent dye. Then we allow the Sanger sequencing reaction to take place. DNA polymerase will use the dNTPs in the template strand to start replicating new fragments of DNA. We then pour the test tube into a single well onto a gel plate and use capillary electrophoresis to separate the fragments by size. The shortest will be at the bottom as they move quickly through the gel. We get different lengths of DNA because when DNA polymerase uses an artificial base in place of a dNTP, replication will stop and a fragment will be created. We are able to determine which base because we tagged each specific ddNTP with a fluorescent dye. Modern day Sanger sequencing uses a laser and detector to determine which fluorescent dye is present in that fragment of DNA. This detector will then relay the information to a computer that will generate a graph with different colored spikes, allowing us to see the order of the bases for this specific gene. We can then determine markers that will allow us to test for and find this specific gene in other genomes.
Through sequencing, we know that Bull A carries the gene that was found to increase resistance to the BRD virus complex. Bull B does not carry this gene. Through artificial selection, we were able to produce offspring that inherited this gene from Bull A, therefore increasing the resistance to BRD. On the other hand, the offspring of Bull B are susceptible to a BRD outbreak. We're Group 12. I'm Lindsay Witten.
I'm Tara Ellens.
I'm Willem DeVore,
I'm Kirk Fisher.
And I'm Clay Pelton.
and these are our sources, and this is the article we chose, and some sources to back it up from our video today. Thank you.